Hello everyone, welcome to today's lecture on tumor immunology. In today's lecture we're going to be discussing the different aspects of tumor immunology, and this is going to be a two-part mini-series actually. So let's get started. First of all, we have to understand that generally infections, be it by bacteria, viruses, other pathogens, they're going to be inducing certain immune responses. So it's not very far-fetched to understand that our cells, generally speaking, are going to be constantly being monitored by our immune system in order to check if there are any problems happening within it. So naturally, if the immune system was responsible for the suppression of the tumor cell formation, then organisms which are immunocompromised are going to be suffering from increased rates of cancer. This is also very important. So by many experiments that have been done throughout the years and the decades that have passed, it has been apparently very clear that people that are immunocompromised are going to be suffering from higher rates of cancer. Therefore, there is something to do with our immune system regarding this. Also, we have to remember that even though cancer cells are aggressive, they are self-tissues. So basically, these tumor cells are not cells that have been induced, or that have been infected, that have been transfected into us. Essentially, they are just regular tissues that are behaving uncontrollably. Next, we're going to be going through a couple of things actually. Injection with irradiated or damaged cells are going to be rendering the organisms protected or immunized from the formation of tumors of a certain type. This goes hand in hand with the example of vaccines, where we're going to be injecting very small doses of certain viruses, for example, and these and our cells are going to be forming immune cells against these, and therefore the next time we are subjected to these types of viruses, they're going to be easily identifiable by our immune system, and therefore we're going to be surviving them easily, and we're going to be having a very quick immune response. So just like these type of pathogens and these type of diseases, much in the same way this acts, we are going to be having the same sort of thing happening with cancer. So in contrast, if we inject viable tumor cells of a different type, the mouse will form a tumor of that type. Also, if we get a vaccination, for example, against HIV, which is not available at the moment, but let's assume we do have that. So if we have a vaccination against HIV and we get this virus, then obviously we're not going to be infected with this virus anymore. We're not going to be susceptible to any complications. But if we get, for example, the common cold, then we are susceptible to complications regarding this virus. So just like the pathogens act, cancer cells apparently act the same. This further proves the point that there is an immune response against cancer cells, and they are MHC class 1 related. Now, another key idea to highlight is that if tumor cells were isolated from a mouse and then re-injected into a syngeneic, that means we have a genetically identical host, then the tumor formation is going to be occurring. And if they were injected into an allogenic host, which is not identical genetically speaking, then no tumor is going to be observed. Next, we're going to be discussing briefly regarding the tumor-specific antigens. Now, this is rather easy, so let's go through the first one, which is tumor-specific antigens. We have antigens that are only going to be expressed. They're only going to be found when the cells are going to be, cancer cells. So they are antigens that are expressed exclusively on tumor cells. We're not going to be finding them on the surfaces of cells that are normal. Those antigens could be unique to certain types of tumors or shared between tumors of the same type. However, what is important to remember here is that they are only present if these cells are cancers. The second type is the tumor-associated antigens. Well, regarding the tumor-associated antigens, these antigens could be available or they could be present on the surfaces of normal cells as well as cancer cells. However, the difference between the two is that simply these antigens are going to be expressed in different proportions, or even they might be slightly different in comparison to what the one expressed on normal cells. Now, regarding the modern classification of tumor antigens, first of all, they are based on the molecular structure and the source of the antigens that are expressed by the tumor cells and that stimulate T cell or antibody responses in the host. So we're going to be explaining that. First of all, the modern classification, we're going to be talking about the tumor suppressor genes, and a very important and a very prime example is the p53. Well, this is weird at first. Now, this is obviously weird because we know that the p53 are going to be suppressing the tumors. So how do we have these p53 genes expressed in cancer cells. It just makes no sense. Essentially, these cells, these p53, are only either going to be completely inhibited when the cell becomes cancerous, or they're going to be mutated. And if they are mutated, they are going to be expressed in larger numbers. So paradoxically, even though that these p53 cells are actually getting expressed in very large numbers, they in essence are not the ones, they're not doing the effect that we know they do, which is inhibit in the sense we know that p53 is going to be pro-apoptosis. So basically it's going to be inducing apoptosis in those cancer cells. Next, we've got the oncogenic products like the BCL2, the BCLXL. They are products that will facilitate the longevity of the tumor cells by suppressing the pro-apoptotic genes. So this is also very important because these oncogenic, the main idea that we have to always know about cancer cells is that they are going to be, removing the possibility and inhibiting the mechanism that is going to be signaling the cell to be undergoing apoptosis. Therefore, this cancer cell, or this cell in particular, is going to be surviving for a very long time. Next, we've got the glycolipids and the glycoproteins. They are essentially components of the dysregulated membranes in the tumor cells. There's not much to it, actually. Others include the mutated normal genes. This goes in hand in hand, by the way, with the p53. Telomerase overproduction, which are peptides, cancer testis protein, fetal proteins like the alpha fetoprotein that is supposed to be only expressed in the fetus, but in cancer cells they are also expressed in adults. So, we already know that these are not supposed to be happening in here, but they do. So this also triggers that there's something going on that is wrong. Next, we've also got the viral proteins, those of the Epstein-Barr virus and the human papillomavirus. This concludes our episode today. Thank you for watching. This has been Ali from B Biology.